Hello, everyone. It's my great honor to speak in this wonderful virtual conference. I'm Yicao from Nanjic University, China. Today, I'm going to share with you our recent work on ester bond containing pathogen surface protein studied by single molecule AFM. Gram-positive bacteria can cause a range of infections, from mild cutaneous and upper respiratory infections to invasive bloodstream and respiratory infections. In the first step of the infection, they have developed a few strategies to ensure strong adhesion against various mechanical perturbations, such as coughing and sneeze. These strategies include the formation of biofilm, so that these bacteria can fight together against large mechanical forces. They have also developed a way called catch bond, so that the surface bond becomes stronger at higher shearing force. Finally, they have evolved the adhesion proteins with intermolecular covalent bond to make the individual protein domains mechanically stronger than the one without such covalent linkage. Intermolecular covalent linkages can bypass the molecular stress applied between the host and the bacteria, preventing these domains from unfolding. Typically, there are three types of intermolecular covalent bonds, including isopeptide formed between lysine and asparagine in SPY 0128, ester bond between threonine and glutamine in CPE 0147, and the ester bond between cysteine and glutamine in SPY 0125. These bonds are formed spontaneously without the need of any external enzymes. The proteins containing such covalent linkages are often found in rod-like pili and other large multi-domain proteins such as microbial surface components recognizing adhesive matrix molecules, MSCR, AMMs, of gram-positive bacteria. It would be interesting to study the mechanical properties of this protein because these studies are of fundamental importance to the understanding of the functions of these proteins as well as the mechanical design of strong protein folds. In the early work from Fernando's group, they use AFM-based single molecule force spectroscopy. They show that the isopeptide bond can mechanically lock the protein structure from unfolding. They made a chimeria polyprotein of I27 and two SPY 0128 domain. When isopeptide bonds are formed, they cannot detect any mechanical unfolding signals of SPY domains. Once they did point mutation to the proteins to remove the isopeptide bond, these protein domains unfold at forces around 300 picnewton. Although the unfolding forces without isopeptide bonds are still very high, they are not as high as the one with isopeptide bonds. Greter and co-workers studied the unfolding of a SPY domain using molecular dynamic simulation. Similar to the experimental studies, they found that without the isopeptide bond, SPY ruptures at around 600 picnewton, while with isopeptide bonds, they did not see the unfolding of a SPY up to 1 nanonewton. In an equilibrium constant force simulation, they fixed the force between the N and the C terminal to 300 picnewton and studied the forces on individual hydrogen bonds between the two terminal beta strands. They found that the force varied dramatically for the protein without the isopeptide bond, indicating the dynamic dissociation and formation of the bond. In contrast, with the isopeptide bond, the neighboring hydrogen bonds are stable, showing small variations on the shared forces. Both experimental and simulation studies indicated that the significant role of isopeptide bond in mechanically stabilizing the protein structures. In contrast to the isoprene bond containing proteins, the thiester bond containing protein typically can partially unfold first to the thiester bond position. Then, if there is no nucleophile around, the carbon-sulfur bond is strong enough to prevent the unfolding of the rest of the protein sequence. However, in the presence of nucleophilic compounds, the thiester bond can be ruptured even through the protein structure is not unfolded. In other words, the thiester bond can be accessed by the external nucleophile, such as amino or thiol groups. Banfield and Schwartz-Linker found that using this mechanism, the thiester containing domains in protein adhesion can bind the host by forming covalent linkage with fibrinogen. Once the intermolecular covalent linkage is formed, they can serve as a mechanically stable covalent linkage. Recently, Alonso, Caballero, and Fernandez showed that the formation of the intermolecular covalent bond is controlled by the force unfolding equilibrium of the protein at forces below 6 picnewton. The unfolding of the protein can drive the dissociation of the intermolecular bond and reform the intramolecular thiester bond. However, at high forces, the protein remained in the unfolded state, thus preventing the reformation of the thiester bond. Ester bond is another type of intramolecular covalent bond formed between the side chain of threonine and glutamine residues. It was first discovered in the surface anchoring protein CPE0147 and then widely found in many bacterial surface adhesive molecules. The CPE0147 protein contains an N-terminal adhesive domain with a surface binding thiester bond and the C-terminal bacterial cell wall anchoring motif, LPKTE. In between, there are 11 tandem repeats of ester containing domains. The structure of C1 and C2 have the crystal structure in PDB. Besides the intramolecular ester bond, there are also 2 calcium binding sites in each domain. Calcium binding do not affect the thermodynamic stability of this domain too much, but may change the local conformation dynamics, as indicated in previous studies. The ester bond in these proteins generally located at the same position as the isopeptide bond in SPY 0128. They lock the terminal beta strands. Note that the 2 proteins show different topologies, and only CPE0147 has the calcium binding sites. As ester bonds are prone to hydrolyze at both acidic and basic conditions, it is largely unknown how such unique chemical properties affect the mechanical properties of ester bond containing proteins. Therefore, we decided to study the ester bond containing protein C1, the first ester bond containing domain from the N-terminus of CPE0147. When we started the experiment, we thought that we should use covalent linkage to pull the C1 domain. In this design, the protein is anchored to the surface through melamide thiol linkage and picked up by the cantilever through spike-catch-spike-tag pair. This interaction pair is exactly developed based on the spontaneous formation of intramolecular isopeptide bond of SPY domain. We also used GB1 domains as the fingerprint, which unfold at around 200 picnewton. in our experiment, we can clearly see the unfolding of GB1 and the rupture of the covalent linkage at forces around 1 to 2 nanonewton. Note that the rupture forces do not correlate to the hydrolysis of the ester bond, because the rupture of the ester bond will give rise to an additional peak due to the release of the trapped length after the ester bond. We then read a very nice paper by Gaub and Bernardi and the co-workers, showing that the non-covalent pair between SDRG and FG beta ruptures at forces above 2 nanonewton, as the non-covalent linkage allowed more single molecule events to be probed in each experiment. We therefore changed the new experimental design using SDRG and FG beta to pick up the polyprotein. Still, we cannot observe the unfolding of C1 and the rupture of the ester bond. Only GB1 unfolding and the rupture of FG beta SDRG were observed. It is surprising that in this design, we can pull the C1 domain up to a force even higher than that from previous covalent linkage. We're not quite sure about where is the weakest part yet. We suspect that this is related to the maleimide style linkage. Nonetheless, this is not the focus of this talk. Next, we mutate threonine 11 to alanine so that the protein cannot form intramolecular ester bond. We engineered the polyprotein and performed the single molecular FM experiment, following the same protocol as the wild-type C1. In this case, we can clearly observe the unfolding of the mutated C1 domain, giving rise to a contour length increment of 47 nanometer and the rupture forces of around 100 picnewton. This indicates that the C1 domain is mechanically weak in the absence of the ester bond. Still, we don't know the mechanical properties of the ester bond. In the Y-type C1 domain, we cannot observe the rupture of the ester bond in the protein. We propose that this may indicate that the ester bond is not exposed to the water molecule. To test this hypothesis, we engineered a circular permutant of C1 by linking the original N and C termini using an elastin-like peptide and create a new N-C termini at a special loop. Then we repeated the single molecule experiment using this circular permutant. In this case, we observed the C1CP unfolds in 2 steps, confirming that ester bond is formed. The first corresponds to the unfolding of the protein after the position of the ester bond with a contour length increment of around 38 nanometer. And the second step corresponds to the hydrolysis of the ester bond with a contour length increment of 23 nanometer. The contour length increments measured experimentally match to the theoretically predicted values. The unfolding forces of C1CP are around 100 picnewton. We also did a control experiment to mutate the threonine 11 to alanine. The protein is almost completely unfolded without the ester bond. More surprisingly, the ester bond is mechanically labile, with rupture force in a broad range and centered at around 100 picnewton. The rupture forces seem to be independent of the pooling speed and also the pH. Changing to acidic pH or more basic pH does not change the rupture forces of ester bond, but increase the chance to observe the bond rupture events from 15% to around 25% at pH 6 and pH 9. We are currently trying to figure out the underlying mechanism using constant force experiment. Nonetheless, our results are consistent with the theoretical prediction from Bolatov and co-workers. They found that the hydrolysis of ester bond in water following a two-step mechanism. The first step involves the attack of the nucleophile proton or OH to form the tetrahedral intermediate TS1, and the second step corresponds to the decomposition of the CO bond under force. They found that although the second step is force dependent, the first step is the rate limiting step under force. Therefore, the overall rupture forces of ester bond show a weak and nonlinear force dependency. The probability of observing the ester bond rupture increases at basic or acidic pHs because the chance to form TS1 increased under these conditions. Note that according to the study by Young and Baker, the formation of the intermolecular ester bond release an ammonia and glutamine becomes glutamic acid. It is unknown whether the ester bond can reform or not. We perform the refolding experiment by picking up a polyprotein unfolding all protein domains, relaxing the molecule to the position close to the surface, and allowing the molecule to refold for 10 seconds and pulling the molecule again to see whether the protein is folded or the ester bond is reformed. Using this protocol, we found that in 11% of the events, we can see the refolding of the C1CP and the reformation of the ester bond. In some cases, we only see the formation of the ester bond, which may imply that the folding of the C1CP depends on the formation of the ester bond. However, we expect that the folding and the reformation of the ester bond of wild-type C1 may be much more efficient than the circular permutant. To understand the molecular details underlying the ultra-high mechanical stability of C1, we performed steered molecular dynamic simulations for both wild-type C1 and C1CP. For wild-type C1 without force load, the ester bond was buried to a large extent and shielded from water attack by the nearby residues. When a constant pulling force of 1.5 nanonewton was applied to N and C terminal residues, the protein structure only partially unfolded at the force-bearing first and last beta strands. However, as the ester bond was located at the force concentration point, it prevented force propagation to the rest part of the protein and made the partially unfolded structure resemble the native structure. As the rupture of the ester bond requires water attack, the structure integrity of C1 wild type can in turn ensure high mechanical stability of the ester bond. In comparison, for circular permutant, the ester bond started to sustain the pulling force only at the later stage of unfolding events. Applying pulling force led to complete protein unfolding and exposure the ester bond to water. To estimate the reactivity of the ester bond in the three conditions, we quantified the number of water molecules within 5 angstrom from the epsilon oxygen atom of the glutamine 141. For wild type, even at a constant pulling force of 1.5 nanonewton, the number of water molecules accessible to the ester bond was only slightly higher than that without force. In contrast, for circular permutant, the ester bond was surrounded by around 9 water molecules due to the unfolding of the protein structure. The solvent accessible surface area show the same trend. Such results clearly demonstrate that mutual interplay between the chemical event and the protein mechanical stability. inspired by the molecular mechanism that allowed the integration of labile ester bond and weak protein fold to achieve remarkable mechanical stability, we hypothesize that it is also possible to use the C1 structure as a mechanically robust cage or safe house to protect other chemically active bonds from solvent accessible molecular attack. To this extent, we replace the ester bond in C1 to a dicer bond through T11C and Q141C mutation and study the mechanical property of this protein in the presence of reducing agent, TSAP. The mutant is named as C1dF. Note that dicer bond was not 100% formed in all proteins. If dicer bond is not formed, it will give rise to a confidence increment of around 47 nanometer. If protein is unfolded but the dicer bond is not formed, the confidence increment will be 41 nanometer. Unlike the case of ester bond, which forms in almost all C1 domains, the dicer bond is only formed in around 90% of total events. Among them, the dicer bond rupture events only account to 40.3% of all events. However, in the absence of reducing agent, this group is missing. Nonetheless, since most dicer bond remaining unruptured in the presence of reducing agent, our result indicates that the dicer bond in C1 DF is also protected. To further confirm this, we studied the mechanical unfolding of the circular permittant of this dicer bond mutant by replacing the ester bond in C1CP2 dicer bond. The predicted confidence increment in that one without dicer bond give rise a confidence increment of 62 nanometer, and the one with dicer bond unfold in two steps with confidence increment of 38 and 23 nanometer respectively. As in this dicer bond mutant, the partially unfolded protein structure cannot prevent the attack from TSAP. The rupture of dicer bond was observed in 42.7% of total events, even without TSAP. In 3.1% of the traces, we observed the rupture of the dicer bond at forces around 2 nanonewton. Taken together, our results indicate that the C1 structure can efficiently prevent the reducing of the embedded disulfide bond in the reducing environment under forces, like the protection effect for ester bond. This result suggests that the unique correlation of the protein structure and the location of the intermolecular bond is responsible to the ultra-high mechanical stability of the protein domain, regardless what type of bond are the force concentration point. This can also explain that the ester bond containing proteins show similar mechanical properties as the isopeptide bond containing proteins with similar structures. A previous bioinformatics analysis suggests that this type of ester bond is conserved in many cell surface proteins of gram-positive bacteria. We hypothesize that these proteins also exhibit outstanding mechanical properties. To confirm this, we studied the mechanical unfolding of another ester bond containing protein PAF from gram-positive bacteria, Pavimonas sp. Like the C1 domains of CPE 0147, we did not observe any unfolding signatures of wild-type PAF up to a stretching force of 2 nanonewton. Once the ester bond was deleted by mutation, the protein unfolded at force around 200 picnewton. It seems that the chemically labile ester bond play similar roles as isopeptide bonds to stabilize the structure of gram-positive surface proteins. Then the question will be why these proteins containing ester bond instead of isopeptide bond. We thought besides strong binding, bacteria also need to develop a mechanism to release the mechanical load to migrate and spread over tissue surface for colonization, especially on wound tissues. The infected bacteria can proliferate quickly and cause inflammation. The wound sites typically show basic pH and low salt concentrations. We therefore study whether the mechanical stability of wild-type C1 is affected by pH and calcium concentrations. We add EDTA to remove the calcium ions. Majority of the force extension curves were still the same as that under normal conditions without showing any C1 domain unfolding or ester bond hydrolysis features. However, we observed that around 18% of events at pH 9 and around 22% events in the presence of EDTA showed clear ester bond rupture events with a countless increment around 41 nanometer, indicating that the structure of C1 becomes flexible under these conditions. As such, they cannot fully protect the ester bond from water attack. Changing pH to basic conditions showed the same effect as lowering the calcium concentrations. With this result, we therefore propose the mechanism that disciplines the stalk region of CPE 0149 may be beneficial for the colonization of the bacteria. The stability of the stalk domain is critical for the force propagation to the tip adhesive. When the IG-like domains of the stalk domain are folded, they can efficiently transduce force to the tip adhesive for strong surface anchoring. When they are partially unfolded, they can release the force on tip adhesion and promote its detachment from the tissue surface for migration. So in conclusion, in this study, we found that CPE 0147 is extremely resistant to mechanical forces. The ester bond locked the structure to a partially unfolded conformation in which the ester bond remains largely water inaccessible, displaying the present structure at basically low calcium conditions, reduce the protection effect and leads to hydrolysis of the ester bond. And we propose that this relate to the infection mechanism. And this work is published in Nature Communication this year. In our group, we not only study the single molecular mechanics and mechanobiology, we also try to using this knowledge to engineer materials of design and mechanical properties, and hope we can show you some results in the future. Finally, I'd like to thank my collaborators and then my students, thank the funding agency for the financial support, and thank you for your attention.